Hello everyone, welcome to a new lecture. It's our lecture 4 and in this lecture we will talk about biological molecules. So firstly I will discuss the objectives of today's topic. In this lecture we will cover the following topics. We will discuss about the biological molecules in detail and we will describe the carbohydrates, fats, proteins and nucleic acids. What are biological molecules? Biological molecules are substances that are produced by the cells and living organisms. Biological molecules are also called as biomolecules. They have a wide range of sizes as well as structures, and they perform a variety of functions. So the four major types of biological molecules or biomolecules include carbohydrates, lipids, proteins and nucleic acids. So carbohydrates, lipids, proteins and nucleic acids are the four major types of biomolecules or biological molecules. Now we will discuss the carbohydrates in detail. So firstly we should talk about what are carbohydrates. So the word carbohydrate literally means hydrated carbons. Carbohydrates are composed of carbon, hydrogen and oxygen, and the ratio of hydrogen and oxygen is the same as in water. The general formula of carbohydrates is CxH2OY. If we chemically define the carbohydrates, then we can define the carbohydrates as polyhydroxyaldehydes or ketones are complex substances which on hydrolysis yield polyhydroxyaldehyde or ketone subunits. So it is the chemical definition of carbohydrates. Here we use a word hydrolysis. What is hydrolysis? Hydrolysis is the breakdown of large molecules into smaller molecules by using the water molecules. Carbohydrates occur abundantly in living organisms. Cellulose of wood, cotton and paper, starches present in cereals, root tubers, cane sugar, milk sugar, these are all examples of carbohydrates. Now we will talk about the functions of carbohydrates. So carbohydrates play both structural as well as functional roles. Simply, carbohydrates are the main source of energy in the cells. So the main source of energy in the cells are carbohydrates. And some carbohydrates are the constituents of cell walls in plants and microorganisms. So these are the major functions of carbohydrates. Now we will discuss the classification of carbohydrates. So carbohydrates can be classified into three categories: monosaccharides, oligosaccharides and polysaccharides. Firstly, we will discuss the monosaccharides. Monosaccharides are the simple sugars. They are sweet in taste and easily soluble in water and cannot be hydrolyzed into simpler sugars. In nature, monosaccharides with 3 to 7 carbon atoms are found. Monosaccharides include glucose, fructose and galactose. So this is the structure of glucose, this is the structure of fructose, and same this is the structure of galactose. These are the ring structures of glucose, fructose and galactose. These three are monosaccharides. What are oligosaccharides? Oligosaccharides are less sweet in taste as compared to monosaccharides and less soluble in water. On hydrolysis, oligosaccharides yield 2 to 10 monosaccharides and a covalent bond is formed between two monosaccharides, and this covalent bond is known as glycosidic bond. So the most common disaccharide or most familiar disaccharide is sucrose. Sucrose is also known as cane sugar, and on hydrolysis it yields glucose as well as fructose. So here we can see that it is the glucose and it is fructose. And sucrose is a disaccharide which on hydrolysis yield glucose and fructose. And a covalent bond is finding between two monosaccharides which is known as glycosidic bond. So glycosidic bond is a bond that is formed between two monosaccharides. What are polysaccharides? Polysaccharides are most complex and most abundant carbohydrates. These are usually branched and tasteless, and they are formed by several monosaccharide units linked by glycosidic bonds. They have high molecular weight and they are sparingly soluble in water. Polysaccharides include starch, glycogen, cellulose, dextrin, agar, pectin and chitin. So this is showing the polysaccharides. As we know that polysaccharides are formed by many monosaccharide units when monosaccharide units are linked by glycosidic bond. So here glucose molecules are linked by glycosidic bonds and they are formed a polysaccharide which is known as starch. So starch is also a polysaccharide. Now we will discuss about starch. Starch is found in fruits, grains, seeds and tubers. On hydrolysis, starch yield glucose molecules. As we have discussed earlier in this structure, that starch on hydrolysis yield glucose, and many glucose molecules when joined together through glycosidic bond they form this polysaccharide like starch. So starch has two types. Number one is amylose and second one is amylopectin. Amylose starch and amylose amylopectin starch. Amylose starch are unbranched chains and soluble in hot water, while amylopectin are branched chains and these are insoluble in hot or cold water. So starch give blue color with iodine. Now we will discuss about glycogen. Glycogen is also known as animal starch. It is found abundantly in liver as well as in muscles. Glycogen is insoluble in water and it gives red color with iodine. So on hydrolysis, glycogen also yield glucose. What is cellulose? So cellulose is the most abundant carbohydrate in nature. It is also a main constituent of cell walls of plants and it is highly insoluble in water. On hydrolysis, cellulose also yields glucose molecules and cellulose gives no color with iodine. So cellulose gives no color with iodine while glycogen gives red color with iodine and starch gives blue color with iodine. What are lipids? So lipids are a heterogeneous group of compounds that are related to fatty acids. They are insoluble in water but soluble in organic solvents like alcohol. Lipids include fats, oil, waxes, cholesterol and other related compounds because they are a heterogeneous groups of compound. What are the functions of lipids? So lipids are hydrophobic compounds and are component of cellular membranes. Lipids are also used to store the energy. Due to higher proportion of carbon and hydrogen bonds and lower proportion of oxygen, lipids store double the amount of energy as compared to same amount of any carbohydrate. So lipids also provide insulation against atmospheric heat and cold and also act as waterproof material. Here is the structure of lipids. As we can see that lipid consists of a polar head which is hydrophilic. Hydrophilic means water loving. While they have the non-polar tails. So the tails are hydrophobic while the polar heads are hydrophilic or water loving. Here is the structure of lauric acid. It is a fatty acid commonly found in coconut oil. Here is a carboxyl group and here is a hydrocarbon chain. What are proteins? Proteins are the most abundant organic compounds that are found in the cells. They are present in all types of cells and in all parts of organisms. What are the basic functions of proteins? Proteins build many structures of the cells. All enzymes are proteins. As hormones, proteins regulate the metabolic processes. Some proteins like hemoglobin work as carriers and transport the specific substances like oxygen, lipids, metals, metal ions, etc. Some proteins called antibodies and they defend the bodies against pathogens. There are some proteins which are blood clotting proteins and they prevent the loss of blood from the body after any type of injury. Proteins are also called as polymers of amino acids. Amino acids are the compounds that contain carbon, nitrogen, oxygen and hydrogen. So the number of amino acid varies from few to 3000 or even more in different protein cells. So proteins can also be defined as the polymers of amino acids. What is peptide bond? So peptide bond is a bond that is formed by a linkage between the hydroxyl group of carboxyl group of one amino acid and the hydrogen of amino group of another amino acid. And in this process, water molecule is released and carbon and nitrogen link is formed, which is known as a peptide bond. As in this diagram we can see that this is a carboxyl group and it is an amino group and these are two amino acids. So when a linkage between the carboxyl group of one amino acid and amino group of another amino acid happens, then a water molecule is released and a peptide bond is formed. So here it is a peptide bond which is formed between two amino acids and it is formed between the hydroxyl group of carboxyl group of one amino acid and the hydrogen of amino group of another amino acid, and a water molecule is released in this process. Now we will discuss about the structure of protein. What is meant by primary structure of protein? So primary structure of protein comprises the number as well as the sequence of amino acids in a protein molecule. What is the secondary structure of protein? In secondary structure, the polypeptide chains usually coiled into a helix or into sheets. What is meant by helix? Helix is a spiral formation of the basic polypeptide chain. So secondary structure consists of polypeptide chain which coiled into helix or beta pleated sheet. Beta pleated sheets are formed by folding back of a polypeptide. So in secondary structure, hydrogen bonds are formed among the amino acids molecules. What is the tertiary structure of protein? Bending and folding of a polypeptide chain upon itself, forming a globular shape. This is known as tertiary structure. So there are three types of bonds in tertiary structure which are known as ionic, hydrogen and disulfide bonds. Now we will talk about the quaternary structure of protein. Polypeptide tertiary chains are aggregated and held together by hydrophobic interactions, hydrogen and ionic bonds. So, hemoglobin exhibit the quaternary structure. Here we can see that this is the primary structure of protein in which the number and sequence of amino acids. This is the secondary structure of protein, and in this we can see that the beta pleated sheet and alpha helix, and hydrogen bonds are formed among the amino acids molecules. And here we can see that the tertiary structure of protein, which is formed by bending and folding of a polypeptide chain upon itself. And this one is the quaternary structure of protein, in which polypeptide tertiary chains are aggregated and held together by hydrophobic interactions and hydrogen and ionic bonds. What is nucleic acid? Nucleic acids are those acids that were isolated from the nuclei, and due to their isolation from the nuclei and their acidic nature, they are known as nucleic acids. So nucleic acids are of two types: deoxyribonucleic acids, which are also known as DNA, and ribonucleic acids, which are known as RNA. So in nucleic acids, nitrogen bases are formed, and nitrogen bases are of two types. Number one is the single ringed pyrimidines, which includes cytosine, thiamine, uracil, and double ringed purines, which include adenine and guanine. So nucleic acids consist of two types of nitrogen bases. One is the single ring nitrogen bases, which are known as pyrimidines, and second one is double ringed nitrogen bases, which are also known as purines. And pyrimidines include cytosine, thiamine, and uracil, while purines include adenine and guanine. What is DNA? DNA is the hereditary material. It controls the properties and potential activities of a cell. Model of DNA. So James D. Weston and Francis Crick built the model of DNA. So they describe that DNA is composed of two polynucleotide chains, which coiled opposite or anti-parallel around each other and form a double helix. So there are two hydrogen bonds between A and T pair and three hydrogen bonds between G and C pair. So in DNA there are two hydrogen bonds between A and T and three hydrogen bonds between G and C, like guanine and cytosine. So DNA occurs in chromosomes, in the nuclei of the cell, and in less amounts in mitochondria as well as in chloroplasts. What is RNA? RNA molecules occur as single strand which may be folded back on itself to give a double helical characteristics. So the nitrogen bases that are formed in RNA are cytosine, guanine, uracil, and adenine. So here the bonds are formed between cytosine and guanine and uracil and adenine. So here in RNA, pairing is between cytosine and guanine and uracil and adenine. RNA is synthesized by DNA in a process which is known as transcription. So transcription is a process in which RNA is synthesized by DNA. Occurrence of RNA. So RNA is present in the nucleus, in the ribosomes, in the cytosol, and in smaller amounts in the other parts of cells. So this diagram is showing the difference between DNA and RNA. So this is the structure of RNA, which is the single strand structure, while the structure of DNA is double strand and it is formed a double helix. So DNA is also known as deoxyribonucleic acid, while RNA is known as ribonucleic acids. And nitrogenous bases in RNA include cytosine, guanine, adenine and uracil, while nitrogen bases of DNA include cytosine, guanine, adenine and thymine. Now we will discuss about the types of RNA. First one is the messenger RNA. So as the name shows that it takes the genetic message from nucleus to the ribosome in the cytoplasm for the formation of particular proteins. So messenger RNA comprises about 10 to 20% of cellular RNA. Second type is transfer RNA. Transfer RNA comprises about 10 to 20% of the cellular RNA, and transfer RNA picks up the amino acids and transfer them to ribosomes, where they are linked to each other to form the proteins. What is ribosomal RNA? Ribosomal RNA make up to 80% of the total RNA, and it acts as machinery for the synthesis of proteins. So the major portion of RNA is made up of ribosomal RNA, which comprises about 80% of the total RNA. Now there are some tests which we will discuss. First test is the iodine solution to test for starch. Add iodine reagent to a solution or directly on a potato or other materials such as bread, crackers or flour. So a blue-black color results if the starch is present. So if starch is present, then the color will turn into blue-black. But if starch is not present, then the color will stay orange or yellow. Benedict's test for reducing sugars. In this test, we use the Benedict's reagent, which confirm the presence of reducing sugar like glucose. Benedict reagent starts out aqua blue. As it is heated in the presence of reducing sugars, it turns yellow to orange. The hotter the final color of the reagent, the higher the concentration of reducing sugar. So in this diagram, we can see that here the color is blue, which shows that no reducing sugars. Here the color is green, which show a traceable amount of reducing sugar. Here the color is yellow, which show low presence of reducing sugar. And it is the orange red color, which shows the moderate amount of reducing sugars. And black brick red color shows higher concentration of reducing sugar. What is burette test for proteins? So in this test, take 3 clean and dry test tubes. Add 1 to 2 ml of the test solution, egg albumin and deionized water in the respective test tubes. Add 1 to 2 ml of burette reagent to all the test tubes. Shake well and allow the mixture to stand for 5 minutes. observe the changes in color. If no color change, then it shows that the proteins are absent and the solution will remain blue, and it is the negative burette test. But if the solution turns from blue color to deep purple color, it shows that proteins are present, and it is the positive burette test. Ethanol emulsion test for fats and oils. Add a few drops of the liquid food sample to a dry test tube. Add 2 cubic meter of ethanol and shake it thoroughly. Then add 2 cubic centimeter of deionized water and make observations. If the solution remains colorless, then no emulsion is formed and liquids are not present. But a layer of cloudy white suspension which is formed at the top of the solution shows that liquids are present. What is DCPIP test for vitamin C? So in this test, the oxidizing agent is dichlorophenol indophenol. It is a dye which is blue color when in oxidizing form and colorless in reduction form. When it is added into vitamin C solution, the vitamin C reduces the dye and decolorizes the dye. Therefore, the decolorization of this dye, dichlorophenol indophenol, indicates the presence of vitamin C. Why water is known as the universal solvent? Due to its polarity, water is an excellent solvent for polar substances. Ionic substances, when dissolved in water, they dissociate into positive as well as in negative ions. While non-ionic substances having charged groups in their molecules are dispersed in water. Almost all the reactions in the cells occur in aqueous media. In cells, all the chemical reactions are catalyzed by the enzymes, and enzymes work in aqueous environment. That's why water is called as universal solvent. I hope you understand the whole topic. Thank you.